For those who dedicate their lives to sporting excellence, there is often no greater prize than winning. This pressure and desire for success can push athletes to engage in the prohibited and dangerous culture of doping. Blood doping includes all means undertaken by healthy athletes, aimed at producing an increased or more efficient mechanism of oxygen transport and delivery to peripheral tissues and muscles. These processes are all related to increasing the number of red blood cells present within the blood. Red blood cells account for 45% of the blood's volume and contain proteins called hemoglobin, responsible for carrying oxygen to the body and returning carbon dioxide to the lungs. Erythropoietin, or EPO, is a peptide hormone that is produced in the kidneys and acts on the bone marrow to stimulate red blood cell production. If an athlete chooses to inject themselves with additional EPO, they are stimulating the production of additional red blood cells, thus increasing the oxygen carrying capacity, allowing them to work harder and longer. EPO has become well known for crippling cycling giant Lance Armstrong of his 7 Tour de France victories. Blood doping through transfusions does not involve the administration of drugs, but is a technique involving the removal of around 2 units of blood from an athlete, which is taken away, stored, and later reinfused into the athlete. The removal of this blood stimulates the bone marrow to replace the lost blood through the generation of new red blood cells. The athlete's blood replenishes to normal after 10 to 12 weeks. Several days before competition, an athlete would then reinfuse the stored blood back into the body. The increase in the number of red blood cells results in higher volumes of the oxygen-carrying proteins, hemoglobin, also present within the blood. The process of doping therefore allows greater amounts of oxygen to be transported to the working muscles, resulting in a higher level of performance without the use of the anaerobic energy systems. It is important to note that this process will not increase the maximum force the muscle can generate, but will permit the muscle to do more work for longer, hereby reducing fatigue. For some athletes, this extra bump can mean the difference between a gold or silver medal, or whether or not a world record is broken. All methods of blood doping are prohibited under the World Anti-Doping Agency's list of prohibited substances and methods. Blood doping control is an essential part of anti-doping programs to promote and protect the integrity of sport and the health of athletes. Doping is ultimately cheating and is contrary to the spirit of fair competition, as the process increases the athlete's ability to go faster, stronger, and for longer.